The last part will be about new targets for coronavirus treatment. Here is an interesting paper published on the preprint website. The authors described a work about a study of protein-protein interaction between novel coronavirus and the human. They conduct mapping to reveal possible drug targets and possible drug repurposing. As you can see, they investigated PPI between 332 human proteins and 26 virus proteins. On this slide, it shows that 3 molecules, such as TMCB and the semiotacitinib, both of them may block CK2 protein. Here, the authors proposed that these 3 compounds could block the interaction between BRD2 and the protein E. As we mentioned in previous slide, the coronavirus use spike glycoprotein to facilitate its entry into human cell. It's quite challenging to block the protein interaction between S protein and ACE2 by small molecules. Usually, the best approach is using polyclonal antibody. However, in this preprint paper, authors suggest furin may work as a potential therapeutic target for COVID-19 therapy. Furin is a pro-protein convertase. Here, we see this figure shows furin may be involved into the S protein interaction. On the lower left part, furin was found to dock onto this putative furin cutting edge of this is the position arginine 685 of the coronavirus spike protein. Based on that, authors advised some drugs, shows here, may function as potential furin inhibitor for COVID-19 treatment. Okay, I should stop here. First, I want to thank people who helped me prepare the presentation PPT. Their names are listed here, especially these 3 people, Sun Xiuyun, Sun Yonghui, Yang Yiqing. I also want to thank my School of Pharmaceutical Sciences and Tsinghua University. Thank you all. Welcome to join us, and thank you for your time.